Hello friends, today I'll be talking about the interphase of cell cycle. First we should know what is cell cycle. Cell cycle is an orderly sequence of events by which the cell duplicates its genome and other contents and divide into two daughter cells. That is called a cell cycle. And the time period required to complete one cell cycle is called the generation time. Here I mention the generation time of certain kind of cells or organisms. For bacterial cell, the generation time is 20 to 30 minutes. And for each cell, the generation time is 90 minutes. And for onion root tip, it is 20 hours. And this onion root tip, it is the best material to study mitosis in laboratory. And the human cell in culture, the generation time for human cell in culture, that is 22 hours. So now I will discuss about the phases of cell cycle. So we'll discuss about the phases of cell cycle. Phases of cell cycle. I keep some facts before going into the topic. Suppose a candidate has to appear on entrance examination. So he has to study for hours, for days, and then attend the exam so that he comes successful. In the same way, a cell undergoes a preparatory phase. Before dividing. We'll take another case that suppose a cell has 40 number of chromosomes. Suppose a cell has 40 number of chromosomes. 40 chromosomes. It divides by mitosis to form two daughter cells, and each daughter cell also have 40 number of chromosomes. Chromosome number, that is the number of chromosome in each daughter cell, would have 40. So how this is possible? So where this extra chromosome comes? So this is possible because the cell, the mother cell, first doubles the content of DNA. Then it distributes the DNA or the genome equally between the two daughter cells. So now we come into conclusion that a cell cycle has two phases. That is, the first is the preparatory phase, and second is the dividing phase. And this preparatory phase is called interphase, and the dividing phase is called M phase or mitotic phase. So now we'll talk about interphase. It is the preparatory phase and it is the non-dividing phase and also called post-mitotic. This interphase is also called post-mitotic phase. So it's the most active stage of the cell cycle because in this phase there is synthesis of DNA, RNA, proteins, enzymes occurs. So the synthesis of DNA, RNA, proteins, enzymes occurs in the interphase. So it is the longest period of the cell cycle, around 75 to 95% of the generation time. And this interphase consists of three subphases, that is G1, S and G2 phase. Let's go to the G1 phase. This phase is also known as the fast growth phase. It is also known as fast growth phase or post-mitotic phase because this phase comes after mitosis and there occurs. So what are the events occurs in the G1 phase? So there is rapid synthesis of RNA and proteins, and there is intensive formation of the cell organelles like chloroplast, if it is a plant cell, chloroplast, mitochondria, Golgi body, endoplasmic reticulum, ribosome, lysosomes, etc. So these cell organelles are required to carry out various metabolic activities, and there is amino acid synthesis for histone proteins. Histone proteins. In this phase, the histone protein is not synthesized, but the amino acid, those are required for histone proteins, are synthesized in this G1 phase, and also energy-rich chemicals like ATP is synthesized. So overall, the metabolic rate of the cell is very high in G1 phase, and this phase lasts for 8 to 15 hours. 8 to 15 hours. So there is a checkpoint between G1 and S called a G1S checkpoint. So here see the diagram. So this is the G1 phase, the longest phase, and here there is the checkpoint, and this checkpoint assesses the extracellular growth factors or mitogens and the intracellular nutritional state. Means the mitogen is a chemical substance. So mitogens, the extracellular growth factor, that is the mitogens, they are the chemical substance that may be a small protein or peptide that induces or encourages the cell to start division, and the lack of mitogen or nutrient can halt the cell cycle, and the cell enters to G0 phase. Any lack of nutrient or mitogen, this growth factor, the cell can enter to G0 phase. And also the cell can take remedial action. If everything goes correct, then the cell again enters into the cell cycle. Now we'll go to G0 phase. We'll talk about the G0 phase. So what happens in the cell in the G0 phase? The cell undergoes differentiation. The cell undergoes differentiation in G0 phase, so that a cell assumes a specific shape and size like sclerenchymatous cell or cholenchymatous cell. But the parenchymatous cell have the ability to again enter the cell cycle, and some types of cells, that is nerve cells and heart muscle cells, they become post-mitotic. They remain in G0 phase when they reach maturity. So these cells do not divide. The nerve cells and heart muscle cells, they remain in G0 stage or in post-mitotic phase when they reach maturity. Now I will talk about the S phase. S phase, it is also called synthesis phase. The main event of this S phase is the replication of DNA. So in the initial phase, the chromosomes contain only one chromatid. But after S phase, the chromosomes would have two chromatids, and these two chromatids are joined at the point of centromere. So if it is the initial chromosome, so it has only one chromatid, and after the S phase, the DNA replication occurs in the S phase, and the chromosomes contain two chromatids. Here, if the initial amount is taken as 2C, then after S phase, the DNA amount would be 4C, but the number of chromosome remains constant. So here is one chromosome, and after replication means the chromosomes now have two chromatids, but it is counted as only one chromosomes. But the DNA amount is doubled in this chromosome than the previous one. And these two chromatids are called sister chromatids. chromatids because the newly synthesized DNA, if it is the newly synthesized copy, so it is the exact copy of this chromatid. So these two chromatids are called sister chromatids and they separate in the anaphase of mitosis. And also another event, that is the synthesis of histone proteins. Synthesis of histone proteins occurs in this S phase, histone proteins, and other metabolites synthesis also occurs in this phase, and this phase lasts for 6 to 8 hours. So now we'll talk about G2 phase. G2 phase. So it is the second growth phase, or it is also called pre-mitotic phase. Here the cell size and volume increases. Cell size and volume increases, and the size of the nucleus also increases due to synthesis of DNA in S phase. And also there is synthesis of RNA and proteins and synthesis of some other biochemicals for spindle formation. There is synthesis of biochemicals for spindle formation. The spindles are required in the mitotic phase, and the organelles like centrioles are also doubled, and mitochondria, chloroplast also divide. The duration of this phase is generally 1 to 4 hours, and there is a checkpoint between G2 and M. So this is the cell cycle, and this is G1 phase, and this is the S phase, and this is G2, and it is M phase. There is a checkpoint between G2 and M, and this checkpoint here, the cell decides whether to enter the mitotic phase or not. This checkpoint ensures the success of DNA replication. If the DNA is damaged, or if replication is not correct, then the cell can halt the cycle at this point. We can graphically represent the different phases of cell cycle by taking two parameters, that is the duration of different phases with the amount of DNA. So this is x-axis, and this x-axis, that is we take the hours, 6, 12, 18, 24, so time in hours in x-axis, and the amount of DNA. If we take the amount of DNA, initial amount is 2C, and it double amount, that is 4C. And in G1 phase, the G1 phase takes about 8 to 15 hours. So this is the G1 phase, and the DNA amount, that is 2C in G1 phase. And when the cell enters S phase, to the end of the S phase, the amount of DNA becomes double, that is the S phase. So now the amount reaches up to 4C, and the cell then enters to G2 phase. And in this phase, that is the G2 phase, here also the amount of DNA is 4C, and when the cell enters into the mitotic phase, again this amount, the DNA, that is equally distributed between the two daughter cells, and the daughter cells again will have 2C amount of chromosomes, and again the cell enters into G1 phase, and the cycle is repeated. So this is about the interphase of the cell cycle. I hope this video will help you understand the interphase. Thanks.